Professor Mark Light obtained his undergraduate degree at Fairleigh Dickinson University, and he's board certified in clinical laboratory medicine. He received his graduate degrees from the Weizmann Institute in Israel in the fields of cell biology and membrane research. He did postdoctoral research in clinical immunology and immunotoxicology at the Medical College of Virginia and the University of Pittsburgh. Lots of good education, Mark. He's a full professor in veterinary microbiology and preventative medicine in the College of Veterinary Medicine at Iowa State University, and he's a pioneer in his field. Today, he's going to speak to us, and the title of his paper is Microbial Endocrinology and Nutrition: Why the Intersection of Microbiology and Neurobiology Matters to Animal Health. Mark, thank you.
Okay. Great, thank you. First, I just really want to thank the committee for the invitation. As you heard, I have mainly a human clinical background. I've been involved in clinical medicine and surgery for many, many years. And about a year and a half, I got invited up to the vet school at Iowa State. And I've been having a lot of frustration with trying to understand the whole role of the gut and the brain and how this works in what's known as the microbiota gut-brain axis for a long time. And finally, we're thinking about, mice are not the end-all of everything. They don't predict everybody out there. And we only have certain access in mice, and we only could do certain things in humans. There aren't many people who want to donate parts of their gut and brain to be sectioned and studied. And I thought, we need to look at species in between. And I couldn't think of anything better when I went to the vet school to see the ability to look at pigs, cattle, all other species, and say if something means something to an animal, means something to, in a way, evolution, it's got to go across many different species. So I made the transition. And so I've been here about a year in the vet school and getting to know now all the different parameters, especially now swine, and actually have a lot of pig work coming up in my own work, and actually have gotten into all things rumen now, of cattle. Actually, I am studying that. But so what I want to talk to you really today about is what I've learned, some of this. And some of the things I've learned already is that the stress experienced by swine is very similar to animals, to other animals, and to humans, and I'm going to bring that in. But the important thing is that although we know a lot of effects of stress on swine gut health, the purpose of my talk is not to review the literature, but is going to be to introduce to you a completely new perspective of how you want to look at nutrition, how the microbiota in the gut is working, how all this may work together and lead then to the brain. And then that affects the behavior of the animal. Now, one of the things is to say that this is one approach. There are many approaches now in trying to understand the microbiota. And so what I'm going to do is structure the talk to give you some conceptual approaches to understanding this very complex environment. I'm going to introduce to you bacteria as you probably were not taught bacteria even recently in school, which is that they are neuroendocrine organisms. They're not dumb bugs, as we're taught, dividing 1 to 2, 2 to 4, and the rest. These are responsive organisms, and they have a neurochemistry to them that mimics your brain. Okay, and I'm going to, the whole time, harp on evolution, because this is important, and again, one of the reasons I've got into the vet field, an evolutionary vantage point. What does it mean for understanding the complex interactions between diet, host, and the microbiota that influence the swine health? And I'll give you some things initially to say here. If you start thinking of these things as a neuroendocrine vantage point and start thinking of the nutrition you feed as neuroendocrine sources for the microbiota to use and then influence the host, you may have a new way to look at how you formulate your nutritive approaches. This whole concept of neuroendocrine bacterial actions has been termed microbial endocrinology. But talking about the microbiota, I have to start as you know, this is an ever-expanding balloon of interest. Everything you read about today is microbiota and the rest. I like to take people, I have a little longer approach, as you can see by how much hair I have, on the field. And what you do is look back to where microbiology started and what we're doing. Clinical needs always drove microbiology from the earliest times. What I mean by is, really when you're talking about when did it need to take somebody's leg off, arm off, if somebody fell in the soil, was shot a soldier, had an infected muscle, at what point is there such a bacterial load that you have to cut off the limb? And so microbiology was clinical microbiology. They had to figure out how many bugs were in per gram of tissue. You're actually looking at plates from the 1910s where they said, oh, 10 to the 5th is the cutoff. If we see that many there in a gram of tissue from a patient, then we have to cut the limb off. We can't keep debriding the tissue. It's not salvageable. Now, where have we come today from that? Well, this is where we are today. And so at one point I've asked Steve to interrupt me at 5 minutes if I keep going too long, because I want to give you a skeptical approach to the whole microbiota field. I think it's warranted. And what you see today, and my field is microbiota gut-brain, is this is what you see encapsulating. Boost happiness by boosting gut health, wide discussion of hype. So I'll be getting to a bit of the hype. How does the gut influence the brain and the rest? Because we're in an ever-expanding balloon. Those of you who know the microbiota literature know that there are holes being poked right now in that balloon. And my feeling is always going to be a collapsing. The question is to know what is, in a way, real. So with that bit of introduction, let me define what microbial endocrinology is. It's basically the union of two fields you don't usually associate together, microbiology and neurobiology. That makes microbial endocrinology. How does it affect swine health? I will go into both disease and behavior based on mouse and human models that have been done. And there has been a lot of this work actually replicated in the pig. I just won't be showing that today from other researchers. Evolution will be a theme throughout. So the thing to bring up is when you see your animal stressed, the one thing you're always taught is that stress affects immunity, then affects disease. And that's the paradigm that everyone believes and had believed. But is that the complete story? So to address this, many years ago, what I did was simply take mice, and those who ever work with mice know they like to fight in the cage. I won't go into the whole thing to spend all the time on this. This is what's known as a resident intruder paradigm. You put an intruder mouse, the resident attacks it, they fight, and basically this is what happens. This guy gives up, it's in the dark, it's not under light, and you look at the brain and what's going on, and what we found out is that this animal's immune system actually was increased, not suppressed. On acute stress, it increases immunity. But, that was back in the late 80s, a big finding. But from an evolutionary standpoint, again, I'm always going to bring evolution up. It makes sense. If someone comes and bites you, because that's the way most human wounds are done in fights, is a bite, and you bite and they inject their mouth bacteria into your wound, why does it make sense for your immune system to suppress and say, I give up, let the bacteria infect my wound and fall over and die? That's not the way it is. Evolution says you're in a stressful situation, you're going to have an infectious challenge, immune system up. And that's what we found. The components of the immune system that are concerned with infectious disease, such as phagocytosis, are increased over 500%. We thought, fantastic, something different than what people say, it's not suppression. But then, when we actually infected an animal perorally with bacteria, here's what we found. Animals that were defeated had a higher amount of immunological competence, phagocytosis, died faster. How can an animal die faster if their immune system is increased 500% in their ability to kill bacteria? And so the question was, does it make evolutionary sense? For whom? At this point, I actually resigned from the American Association of Immunology and actually went back to microbiology. I'm not kidding, that's what happened. And I reconsidered stress, immunity, and disease, but forgot about immunity. The reason is, when you see a few bacteria, you swallow, let's say, contaminated hamburger meat, and you swallow 10 E. coli 0157. They are damaged bugs. They are not happy bugs. They got to make it through your stomach acid. They find themselves in your small intestine, right? How many survive? 4 or 5. Infectious dose, we know. How do 4 or 5 bugs in the middle of trillions of bacteria actually cause disease? Your immune system will never see them initially. They have to survive in a complex environment. It's the poor bug. You guys, you have to remember about the poor infectious bacterium. Whose survival are talking about? Can the bug, when you ingest it, all of a sudden figure out where it is, use its environment for its own advantage to survive? So, with that in mind, I'm going to make a little detour into nutrition, because this is important to understand. If we're talking about bugs as neurochemical organisms. The thing is that we have to understand that neurochemistry is not restricted just to people and mammals. If we look at foods, for example, we find that bananas contain amazing amounts of neurochemicals, such that if this was a cardiology audience in the 1950s, I would be advising you not to allow your patients to eat bananas because it has so many neurochemicals. The trouble was, back then, what they had done was take whole bananas, blend them down, and found these neurochemicals that would affect the heart. It wasn't until 1959 that Underfield, in Science, this is when you could publish in Science by simply peeling a banana. And that's what he did. He peeled a banana and analyzed the peel versus the pulp. Did he find all the neurochemicals are in the peel, not in the pulp? So it became okay to eat bananas again if you're a cardiology patient. No one explains why, as a banana browns, it accumulates more pure neurochemicals. These are not like what you have in your brain and your body. These are exactly the same. No one's ever explained why a banana has that. Tribal pulses. You've all watched the National Geographic specials, where they beat these seeds onto hot stones, make a bread flour, and then eat it. Back in the 50s, when a lot of the tourists were going there, which were British tourists, they were coming back with cardiac issues. They could not understand what was going on until they found out what they were eating. They actually looked at the tribal pulse seeds and found 8 grams of pure L-DOPA in the seeds per 100 grams of flour. They thought that was impossible. It's super pharmacological. It's what, of course, is involved in Parkinson's. What is a tribal pulse doing that? And people thought, doesn't matter. If you're a neurochemist, even today, you don't care. You say, I heat the bread, I cook it, and I destroy the neurochemical. Because in the lab, it oxidizes readily. But just like, I like to say, Keanu Reeves in the Matrix, that he can get lost in the Matrix, neurochemicals are always protected by the matrix of food, such that they autoclave food for 2 hours with L-DOPA. It retains 98% biological activity. It does not destroy it. So it remains in the food. Other foodstuffs, if you're clinical and you prescribe antipsychotic medicine to patients, you put them on monoamine oxidase restrictive diets, because tomatoes have dopamine, tyramine, cheese has tyramine. So what I'm trying to tell you is neurochemicals are all out there. Potato plants have neurochemicals. They contain norepinephrine and dopamine, just like your stress response. Even common feed based on cereal grains like corn can lead to elevated levels of catecholamines in the brain. So, with that in the background, is it surprising bacteria, when people began looking at bacteria, do they have neurochemicals? And the answer is yes. For example, GABA, gamma-aminobutyric acid, which is the primary inhibitory neurotransmitter in everyone's brain, is found not only in a number of infectious pathogens, but is found in anybody who eats yogurt and eats lactobacillus in their yogurt. They're eating GABA, because those many of them make GABA. Why? We don't know. We have ideas in terms of pH, but they have GABA for other reasons. Catecholamines, thank you. Catecholamines are made by E. coli. The complete biosynthetic pathway you have for stress that you start from L-DOPA, dopamine, norepinephrine, epinephrine, that you make the same coenzymes, the same reaction series, the same kinetics are all in E. coli. No one to date has explained why E. coli has it. So all of these things have neurochemicals. Protozoa have it. Since this is a nutrition audience, I'll ask you, and since it's a swine audience, why does a pig hunt a truffle? You all know pigs hunt truffles. Why? It's a swine audience. They want to have sex with the truffle. You know that. The truffle, and it's a female pig, the truffle contains androsterol, which is in what? Which is in the foaming saliva of the male pig. And so the female has a riding position, if I'm doing this right for swine, I hope, and that's what they train dogs to. So and sometimes people look, wow, what's a truffle doing with the pig's sex pheromone? but the truffle came before the pig. So we don't know why. So the question is, so all this is nice, but do neurochemicals actually affect bacteria? And I like to show the students, and this is actually the first one that was ever done in 1992, where we took Yersinia enterocolitica, we put it in a very hard media for bugs to grow in, because the idea was, don't make it easy for the bugs to grow. Make them work, make them see signals and see if they respond, even in media that is not, nutritive. And what we found is if you grow the bugs and you try to grow them without supplementation, this hard gut type media, they don't grow very much. If you put epinephrine in, they hardly grow at all, because epinephrine is not found in the gut, actually. I won't go into why. You have norepinephrine, but look what happens when you put Yersinia enterocolitica, which is a gut pathogen, into this media. It explodes over 10 to the 8th fold more growth in 24 hours. It's explosive. And if you then, we did experiments where we just simply mix the bugs with norepinephrine ahead of infection, because it's well known in mice, if you try and infect mice with Yersinia enterocolitica, it doesn't take, it won't go. Here's an animal which we put 10 to the 6 bugs in, clean gut, as well known. Simply take a few less bugs, mix them with norepinephrine a few hours ahead of time, and then put in the animal. Huge invasion into the gut. Formation of all these bulbous areas here. So norepinephrine was 50,000 times more effective before, because of norepinephrine. And we thought, great, we're the first ones. And I always tell students, if you think you're the first, you're not the first, because you just didn't do your homework and look in the literature. And you go back as far as 1928, 1929, it was already described in the Comte de Rendis that if you mixed catecholamines in a syringe that had been contaminated and injected into a patient, you can kill that patient with Clostridium perfringens, gas gangrene, in 6 hours. They didn't know why. And that's a whole story in itself on medical technology and the rest, but that was shown. So this has been an observation observed for decades. So, but why? They all thought it was because you suppress the immune system, not a direct link between the immune system and neurobiology. So what I want to bring up to you is that there is an evolutionary relationship between the host. So telling you that catecholamines are in E. coli, what does that mean? And we have the same pathway. It means that our acquisition, animals' acquisition of all of these neurochemical signaling pathways that modulate your behavior, modulate systems, was acquired by late horizontal gene transfer from bacteria. So bacteria had these, we acquired them, your animals acquired them because of late horizontal gene transfer. So microorganisms such as those in the gut and really everywhere don't simply rely on nutritive sources for their survival and energy. They're relying on also neurochemistry, not only their own, but what you're feeding them in your diets, and also what they're getting from the host. It's bidirectional, as I'll go into. It means that they are interactive players the whole time, such that if you, from host to bacteria, the host can produce neurochemicals which then affect the microbiota, affects things like neuroimmunology. You could change this with prebiotics and probiotics. It's in the booklet, so I'll just skip through this a little bit quicker. The same way as microbiota and probiotics make neurochemicals that you feed to the animals, they can then affect host neurophysiology and host neuroimmunology as well. So there's an interaction going which is bidirectional. The host makes neurochemicals that goes into the gut. The bacteria making neurochemicals that we're not even sure what they're doing or where they're going yet. Now, what is any showing that this is real? So here you have pigs, and what I want to show you is that each of these conditions in terms of infectious disease is applicable if you use microbial endocrinology. So Campylobacter and Salmonella in pigs, Staph epi in humans. So people on the clinical side, when they have IV catheters in, hips, replacement, all those things, the most common infection in the surgical intensive care unit is Staph epi, Staphylococcus epidermidis, your skin, your bugs. And they could not understand for decades why they should infect. If the nurse or the person does their job, cleans off very nicely, why is Staph epi getting in there? And for years they thought, well, let's coat the things in plasma, let's do all these things, nothing worked. It wasn't until 2003 when we published this in Lancet, which was the cover at the time, showing that it was the physician doing the harm. Everybody forgot about the bag of material that was hung into the patient. So the way they keep patients' hearts and kidneys going in the intensive care unit as well as elsewhere is the use of catecholamine inotropes to keep heart function and kidney function going. And we simply said, well, maybe it's the stuff that's coming down the tube, which is a catecholamine, is stimulating the bugs to grow. So we simply took what was in the tubes, coming down as medicine, mixed it with staph epi at a concentration of about 3 or 4 bugs per ml, incredibly low. And what you see here is a IV catheter treated that way. That's just background noise. If you put the same neurochemical that's flowing into the tube in the patient, that's what you get, which is an incredible biofilm with all the channels, all the hallmarks of antimicrobial resistance. That to this within 24 hours in a media which is highly restrictive to growth. So it's not LB or any of those who are microbiologists out here. It's a very restrictive media, and that happens. So low-hanging fruit. Again, are you, in a way, the first one doing it? What do we know about the neuroendocrine environment of the gut, especially in swine? Well, we know that it produces neurochemicals, that they must be out there in the gut. But is there any evidence, again, in the literature this has been seen before? And if you go back to 1976, they actually found that Campylobacter, which infects poultry and swine as well, is very difficult to grow, as we know. Microaerophilic conditions in the lab we need to treat it special. But it never makes sense, because here's a bug that is so prevalent, why does it need such special growth requirements all the time? Well, what they did was, this group, was that they took it and they put in norepinephrine here. Here's a plate without it. Here they struck in a low concentration norepinephrine, and they did this in the air, no microaerophilic anymore. And all of a sudden the bugs start growing. They increase the concentration of neurochemical, and here's what happens. In other words, they made the bugs go from microaerophilic to aerobic, simply by giving them a neurochemical. In other words, they become more infective because now they can be transmitted easier. So this was 1976. Again, the connection not being made. Why is this happening? So I'm going to kind of switch gears now, because in the gut is where the action is occurring. So many people don't realize that in your gut and in the animal's gut, the production, metabolism, and neurochemicals is the greatest. 50% of all the body's neurochemicals actually are produced by the mesenteric organ. And this has been shown in swine, in humans, in dogs, all across the board. Because when they started looking, they thought, it has to be the brain, it has to be somewhere else. Why the gut? That's where they are. Your stomach is a dopaminergic organ. You produce over 50% of the dopamine in your body from your stomach, and nobody has yet come up with a reason why. But your body does that. So within the lumen of the gut, you're making neurochemicals. Anybody went to the, not to be gross here, anyone went to the bathroom this morning? Just evacuated out serotonin. Because you're pumping serotonin into your gut, into the lumen of your gut in large quantities for a reason nobody understands, but you do. So norepinephrine and dopamine come out, and dietary sources. So not only is the host producing it, but you're eating it with food. So with all that in mind, putting all that together, we're going to introduce, in the remaining time, the microbiota gut-brain axis, saying that food-derived substrates and neurochemicals are coming in the diet. That's affecting the behavior of the microbiota in the gut. That then, those microbiota could either produce neurochemicals and have an uptake into the portal circulation or interact with the enteric nervous system. Now I'm going to spend a minute on the enteric nervous system, because that is something that you're not taught in school. That then feeds directly to the brain, because your enteric nervous system hooks to your brain, in your pigs, in humans, everywhere. Okay? Behavior and cognition then results, and that I've proposed and others have proposed now, and we're having the initial evidence, feeds back and tells you what you want. So in other words, the bugs are feeding into the loop saying, what do you want to eat? What should I want to eat? And we propose that this is a complete feedback loop. Now, correlation and causation. Just because something happens at the same time something else does, this is a hallmark of microbiota research, it doesn't mean they're related and causative. You have to prove this. You have to go into each step to prove it. That has not been done. We want to believe it's been done, but it's not. This is where the future lies. But it's not been done. We have strong correlative evidence, but that doesn't mean causation. Okay, the enteric nervous system. You'll be happy to know that the vast majority of your surgeons, human side taught, don't even know about the enteric nervous system, although it was the first part of the nervous system by dog yell ever described in the late 1800s from dogs. So your gut has an extensive nervous system that contains over 500 million neurons, which is as much as your spinal cord, and it's directly linked to your brain via a number of pathways, but mainly the vagus nerve. So, and your gut is differentially innervated by this enteric nervous system that then through these vagal pathways hits the brain. So actually physicians and surgeons are not taught about this. This is about 1 or 2 pages in a whole book, but yet they cut the nerves all the time and they don't know what's happening. Now, in the swine-gut communication, this is actually going on because as I was showing you, these nerves go right up to the tips of the villi. They are acquiring information from the lumen and they're transmitting it down, and a lot of the neurons are concerned with obviously gut function, but 500 million neurons was put to me by one of the neuroanatomists I talked to years ago. Aren't there all to move the poop along? They're communicating information, and they'll show you all the way up to the central nervous system. Social stress in the animal can affect this. It's a bidirectional way, the way this works, but it's to the tips of the neurons where they are acquiring information. And here you can see actual pictures of how extensive this innervation is. And this is only 2011. Now the question is where all this information is going and bidirectionality. So let me just show you how these are really related. This is not just my kind of making this up as I go along. It is just last year, this is the incredible part, just last year that this group in general clinical investigation showed this. They took a neuron from the brain, a trigeminal nerve. They took an endocrine cell from the gut. They simply put them down in a petri dish. Do what you want to do. And here's what they wanted to do. They made a nerve. They connected up. They actually, a brain cell put next to a gut cell makes a nerve to communicate with each other. Now, what does that really mean? Is there really stuff to prove this? Well, again, just this should show how recent this material is. 2 years ago in Germany, what is the bottom-up information? Well, one way, I don't know if you've ever seen this, this is an elevated plus maze. This is how all psychoactive drugs that are used clinically start. You take a little mouse, you put it in this little open area, and you say, are you a brave mouse and you want to go out on an open arm and look around? Or you want to stay in the closed protective areas. Everyone who has ever dealt with mice in the garage know that they go to closed, dark areas. They don't like being out in the open, brightly lit areas. If you could make a mouse go from a dark area to a light area, what do you have? You have an antipsychotic agent because now you have an anti-anxiety agent, an anxiolytic. You've destroyed anxiety. You've made them go from dark to light, to an open area, okay? So with that in mind, what did they do? This is the way a mouse tracks anxiety-like behavior. It tracks in the closed arm. But if you simply cut some of those nerves in the gut, here's what happens. Animal all of a sudden starts going in the open arms. In other words, just cutting a few nerves in the gut that communicate with the brain changes the whole behavior of the animal. So the take-home message, and this actually was published, the take-home message is that, they wrote, it emphasizes the important role of afferent visceral signals in the regulation of emotional behavior, AKA, as everyone's mom has always said, gut feelings. You have a gut feeling? They actually wrote, here's the real, in 2015 they proved it. So where we come from? Well, we have an emergence of the human microbiome. What does it mean for the microbiome gut-brain axis? Very quickly, because I can see the time. Okay, good. Just hit me at 5 minutes.
I will.
Okay. In 1911, you're in nutrition, Kellogg. You all know who Kellogg is. Kellogg, yes, the same guy who's Kellogg, good for your morning breakfast. He was a surgeon. What did he have? Clinics. Women, of course, were very flighty then. They were very high stress. No, not being joked. Well, this is true in the regard that husbands brought wives to his clinic because of this. He had a solution, which was to cut out their colons. You think I'm joking, this was an accepted medical practice. And here you see in JAMA, Journal of American Medical Association, in 1911, Kellogg said to colectomy to prove human temperament, meaning female temperament and health, should the colon be sacrificed or reformed? I have labored constantly and earnestly to devise and perfect methods for changing the intestinal flora. 1911, he cut out women's colons. It was a practice for the next 30 years. They didn't do too well. From 1914, in the Medical Journal of Surgery, the control and mastery of man's diet is readily accomplished, but mastery of his intestinal bacterial flora is not. There are cases that present malaise, lack of ambition, every effort in life is a burden, mental depression, bordering on melancholia. A battle royal must be fought, and when this first great struggle ends in a victory for Bacillus bulgaricus, it must be kept on the field of battle forever at guard. If you ate yogurt this morning, you ate what Stowe said. You ate bulgaricus usually, okay? So this is not new. And now we've progressed to midichlorians. You mentioned about religion before. This was actually a peer-reviewed paper. You should look it up in 2014. It was the biomem hypothesis saying that the reason you do a religion is because of your gut bugs. They are determining your religion. So I'm going to stay for the rest of the few minutes left between these two poles on the talk. And to show you that bacteria can affect behavior, I'll show you the first experiment that was done, in which we took bacteria in the gut, they're seen by the brain. So you take Campylobacter jejuni, which is a pathogen, but if you give it to mice, they don't care. It doesn't cause an immune response, it passes harmlessly through their gut. Divides though, this is the important part, it's an active dividing cell. And what we did is we put this into the animal via perorally and measured it on that elevated plus maze that I just showed you before. And what we saw is that simply putting a bug in that passes harmlessly through the animal, you get anxiety-like behavior. And what you actually see are neurons in the brain, in the nucleus solitarius, in the PVN, paraventricular nucleus. This is CFOS staining. So if you see black dots, it means that the neuron's been firing. And you see all of these dots firing, showing you that the bacteria passing through the gut harmlessly, the brain is aware of it. There is communication going on. And behavior results in the term of anxiety-like behavior. Like behavior because anxiety is a human trait. We don't know really what the animal is feeling. So, and I'll finish up with nutrition. So these interactions mean a long evolutionary pathway where all of our neurochemicals first involved in bacteria. That symbiosis means the gut and the brain can be viewed in a new paradigm. Then you have new nutritionally based mechanisms can be identified and new therapeutic approaches, such as prebiotics, probiotics, for example, formulated. The question is whether nutrition could be used as a means to alter microbiota and influence the production of neurochemicals. Would that approach have consequences for the host, considering, let's say, housing conditions? If you can and manipulate this axis, you could come up with a spectrum of new nutritive approaches to benefit the animal that uses this microbial endocrinology as a pathway. So is it possible? Well, the first experiment was using meat. People forget mice love to eat meat. They don't eat a plant-based diet that we feed them in the lab today. It is artificial. So mice were fed meat initially in mouse houses back in the 20s, 30s, 40s, and 50s. So all that we did was we fed mice meat. They loved it so much that they actually crawled into the food containers and broke off the tops. And what happened? Well, what happened was we asked, does that change their memory by eating a meat-based diet, and does it change their microbiota, and does the whole axis change? So for this, we did a whole board design, which is basically to test memory and say, we're going to bait certain holes in a whole board on a flat surface with a treat and see if they remember the treat after a couple of weeks. And just to show you, if you feed meat, you actually increase the diversity of the bugs that are in the gut. This is old 454 pyrosequencing data, if anybody I could talk about later. It just shows you that they're different, that the bacteria in the gut is very different if you feed meat. And what happens is, because you feed meat, we published back in 2009, you increase the memory of these animals. They remember where their treats are. Just by changing the bugs, by feeding a nutritive approaches, they have better secondary memory. I tried to get the cattle people interested in this, submitted grants. They threw them back right away. What are you talking about? They were scared of what I was doing. So it's a correlational study, not direct cause and effect. So with nutrition, the question is whether nutrition can be used as a means to alter microbiota. With that approach, as I said, a new spectrum. And one thing we've tried, for example, has been resistant starch. It has altered the microbiota. We recently published in animals This is normal cornstarch to all these different types of starch. It's all pretty colors, but it just shows you that all of these taxa were changed. We changed neurochemical production in the gut. L-DOPA, glutamine, we measure a spectrum of neurochemicals in the gut. And it changed behavior of the animals. It induced anxiety. Okay, I'm going to skip over probiotics. I have
7 minutes.
Perfect. So just to mention that probiotics make large amounts of GABA. And you all have heard of functional foods, correct? So the question is, can probiotics which make a neurochemical actually affect a host? Well, the work was actually done in Japan a few years ago with Lactobacillus brevis. They took strains of bugs from fish species intestines. Why Lactobacillus in a fish intestine make GABA, no one knows, but they're doing it. Again, all these areas to research for students. Why are they doing it? Either way, they isolated these bugs that make a high amount of GABA from a thing, inoculated them into black soybean milk, made from homogenized beans, so now the milk had high amounts of GABA, and what did they do? They gave them to rats that were made depressive-like. So to treat depression in humans, we do a test in rats, which makes them have a depressive-like condition. I won't go into it right now. But what you then do is feed them what you think is a good drug that will help in humans. And what you do is feed them and see if you could, you hang them and you see how much they want to jitter, just by the tail. It's a tail assay, basically. It doesn't hurt the animal. And you see whether the drug changes that. And here's a control, how long they were immobile, so they were depressive. Then here is F is fluoxetine, which is the drug that's used in humans to treat depressive conditions. And it decreases the immobility, so the animals are not in a depressive-like state. Well, guess what? If you gave them GABA soybean milk instead of the drug, they did just as good, if not better. It's as effective as fluoxetine as an antidepressant. You don't want to have the pharmaceutical industry hear that. Look at that. So it works. I won't go into how we test, but I'll spend the last 5 minutes on this, the skepticism. I feel I have to. This is just in the New Scientist, just out in May of this year. They call it microbiomania, the truth behind the hype about our bodily bugs. Now, I'm not saying that the microbiome is not real. It's real. I just want to address parts that you may not be aware of as you may get into this field. In this article, it said there's a gap between the promise of the microbiome and the current science. It has been filled with misconceptions, snake oil, and hype, microbiomania. Microbiome research is incomplete. We simply do not know what many microbial genes do. I can't harp on how correct that statement is. Good thing, for those doing mechanistic-driven science and appropriate translation models, there'll be enormous opportunities. Why? This is what I call what we think we know and what we really know. So what does it say? What I want to say is that we unite, typically in microbiome research, in a bioinformatics approach, sequencing, and we unite metabolomics. Sequencing, I don't know how many you're familiar with sequence and metabolomics. Metabolomics is done by mass spec and the rest. The thing about mass spec and sequencing, where we get all these sequences, and we unite them to make these interconnected pathways that you see in Science, Nature, and PNS, look great. They're from huge groups and all the rest. They make these connected pathways, tell you what's happening. But there are challenges to this. And the challenges are that in this that you see here, the sequencing, it's millions upon millions of reads. Technologies and platforms change from 454 pyrosequencing to Illumina MiSeq, HiSeq, PacBio, all these things. And you ask the question, well, I've generated all this data from all these platforms. Do they speak to each other? No. How good are these data that I do with sequencing? If I take this sequencing, this sample of my poop, and I split it in two, and I send it to one place and another place, will I get the same answer? Well, if you're doing a blood glucose on a patient, you want to know it's 40 or 400. That depends on whether the person is in shock or not, okay? So you don't want to know that it's 40,000 one time and 4 the other time from another lab. Well, guess what with sequencing? They don't agree, the labs. And this is a well-known issue about sequencing, that if you send it out to different places, they don't agree. They might not agree in the same lab from 1 day to 2 weeks later that you sequence. What does it mean about longitudinal studies? There are huge issues with sequencing itself. It is not like measuring a chemical like glucose. You need to be aware of this. Metabolomics. Metabolomics is an interesting one because I've spent 4 or 5 years and thousands of dollars on it, and it is a frustrating thing because we only know when we get thousands of peaks in an untargeted assay, as you say, where you generate thousands of peaks, we only know what 4 or 5% of all the peaks really are. They are judged by the machine. There are no standards in those machines that identify all 4,000 peaks. They just said, where's the time here that it's coming out? Bang, that means it must be this. It doesn't mean it's that. And it's semi-quantitative or qualitative at best. It doesn't give you absolute units. So now what happens? You take these two technologies and unite them together and you put them through bioinformatics. Don't get me started on bioinformatics. It'll make it really too late. There are so many bioinformatics platforms, it'll make your head spin. And each of them have their own issues. And to give you an idea, 2 weeks ago in MBio, there was a publication out. Someone made the comment about my bacteria making me fat. It was a re-looking from Michigan at all of the data, run through another way of doing it, and they knocked out one of the foundational pillars of the microbiome 2 weeks ago. They said there is no evidence at all yet that the microbiome in humans affects obesity. All the data wasn't analyzed correctly. It's not there. They're saying it doesn't mean it's there, but it may not be there. And don't forget, everything you measure is poop. Poop is what your body didn't want. The real issue is what is in the GI tract that are near nerves, that are up in the areas that then can get into portal circulation or interacting nerves. How are you looking at the same bugs? You can't just take a poop sample and say, hey, I know what's everything in the gut. You know nothing about biogeography. What's known? Very, very little. And just give me an idea about the computational challenges. People say, let's go to whole genome shotgun sequencing instead of 16S. I know I'm throwing these terms at you. So this person in bioinformatics took a tube of water, put in 11 known bacterial species, 11 known ones, mixed them up, sent them away for whole genome shotgun sequencing, should be better than 16S. Got it back, put it through all of the programs, 30 or so programs that are out there, and you know what the programs predicted from those 11? There were dozens, this is a quote, dozens to hundreds of species were falsely predicted by the most popular programs. What does that say? Not very good. And just last month, NIST, the National Institute of Standards and Technologies in Washington, held a meeting just on this topic, the problems that we're having. We need standards. We need to get through this in all parts of the whole pipeline. So it's not as much as who the players are and the numbers. Microbiome V1, your taxonomic approach to saying who's there is dead in the water. It's been done. It's all correlation to disease state and the rest. It hasn't really panned out. We're in microbiome V2 I'll finish with. V2 means it's not who the players are, it's what they're doing. What are they doing? In other words, what are the mechanisms? So back to culturomics. Believe it or not, there's a new term out there, omics. Culturomics, yes, it's old bacterial culturing. A group in France has inputted culturomics, and they've been very successful at it. So to get things, they call it culturomics. So where do we need to go? We need to understand the limitations of the platforms, avoid a washing machine approach, and P less than 0.05 is the holy grail. Realistic animal models that provide translational real evidence to humans, that's where I'm at. That's why pigs, not mice. I'm getting more and more into putting pigs in my grants, have the closest anatomy. And as more than ever, it's always been mechanism, mechanism, mechanism. The days of simple correlation are over. We have entered microbiome V2. Thank you.
We have